So I'd like to introduce Dr. Peter Fox. He's director of the Research Imaging Institute and professor of Radiology, Neurology, Psychiatry, and Physiology, really showing how multiple interests combine to do what we do. At the University of Texas Health Science Center in San Antonio, he's a longtime editor of the journal Human Brain Mapping. He's also originator of the Brainmap project, which includes the Brainmap database and a suite of tools such as Teladoc Daemon and so on for coordinate-based meta-analysis of neuroimaging data. Looking on Google Scholar, he has approximately 50,000 citations to his work and an H approaching 100. And he's one of the first serious imagers that I know of who has combined his work with brain stimulation for many years now, actually, and mostly using transcranial magnetic stimulation. And I'm sure he has a lot to tell us about these topics. So the title of his talk today is TMS PET Studies of TMS Aiming, Network Modeling, and Parametric Modulation. So join me in welcoming Dr. Peter Fox.
Good morning. My thanks to the organizers, to Vince, for putting this together. Definitely a timely and much needed meeting. Which does which? Good. Okay, so parts of what I'll be showing you, I won't specifically flag them, but parts of the intellectual property that I'll be going through are patented or copyrighted, or both, by the University of Texas System Board of Regents. And most of that has actually been licensed, and the licenses are to companies that I do have a financial interest in. So I'm hoping I can do as good a job as our prior speaker in showing science rather than propaganda. It's really quite exceptional. I'm very impressed. So for TMS, one of the things that we began studying a good number of years ago, 1996, was orientation effects. With the first TMS papers, the coils that were being used were round and really didn't lend themselves to studying orientation effects at all. However, pretty soon thereafter, so in the early 90s, B-shaped coils were introduced, and there are different varieties, figure 8, B, D, different coil shapes. And the point of these, though, is all the same, that with a counterphase current, you're having the current dissipate when it goes out and some when it comes in, so you get a short, straight current vector, which is ideal for aiming, right? Assuming that you want to aim something straight rather than something round. So that's good, and as soon as those were introduced, two studies came out, and it's kind of entertaining to look at them. They both used exactly the same stimulus parameters. So I don't know which lab had the mole, but this cannot be a coincidence that one lab was spying on the other for sure, because exactly what they did was the same, and they were published within months of each other. So at any rate, what they did was they aimed straight at the nose as 0, and then 45, 90, 135, all the way around the clock. And both found exactly the same thing. They were using EEG to measure the effects. And what they got was when they pointed at the nose, 45 degrees, they got the maximum MEP. And 90 degrees off of that was the worst. And coming back around at 90, It was reasonable, but they were using biphasic coils, so that's a little hard to interpret. But at any rate, they found identical results. And so what did they make of this? So what they made of it was substantially colored by a fairly influential paper that came out a few years earlier by Day. And this was based on really scholarly work looking back over many years of electrical and magnetic stimulation, mostly in animal models. And so some of the comments that they made in terms of electrophysiology were spot on. They were just spot on. So, for example, the first agrees exactly with what was said about TDCS, is that you're really stimulating pyramidal cells, and it's current along the axon that is really what you want, along the gradient of the body, so along the column. So a voltage gradient parallel to the long axis of the neuron is what you want. It's like, yes, there's a lot of data supporting that. Okay, but the next one was specific to, or aiming more at TMS, and that is, they argued that the closer it is to the stimulating electrode, the more likely it'll be to excite. So although they said orientation in the first principle is really important, basically what they're saying is that it's overruled by intensity. So it's super important, but just not important. And so you really need to deal only with what's closest to the stimulating electrode. And so in the third one, that accordingly magnetic stimulation should hit the gyrus, so right near the cortex. And if it does that, the pyramidal cells are all oriented this way. So that means you can't hit them, because the current's going to be running this way. So although they said they really want to stimulate along the long axis, then they flipped and they said, really, so that what that means is we must be hitting horizontally oriented neurons. It's like, okay, so the horizontal interneurons are all inhibitory and very short distances, like a millimeter is long. But nevertheless, they predicated their predictions on that. And so both the papers interpreted their results, which really said they're stimulating M1. They both said we're stimulating M1, and they both acknowledged that they couldn't reach M1 because they could only hit the convexities, and so they each postulated different mechanisms for how they were hitting M1 when they couldn't hit M1. All right, so that's super convoluted. Why would they do that? But that's kind of where it stayed, and that was the interpretation of these orientation effects, which are really robust, but I believe profoundly misinterpreted because of this early bias that you can only hit what's near the electrode, and that's it. So in 1996, after agonizing about whether you could put a TMS coil in a PET scanner, because there were papers saying that you will kill your PET scanner if you put a coil in it. And there were devices you could buy to protect your PET scanner from the TMS. And so we modeled that and decided that that was just nonsense, even though the company selling the protective mu metal shield was lobbying each MRI to say that if you didn't buy their shield, you'd void your warranty. That's a serious conflict of interest. So as you see here, we've got no mu metal shield, and we modeled the heck out of it and scanned a phantom with the TMS in it, on and off, to see if we could measure any effect. And the answer is we couldn't measure any effect on the PET scanner. So that was a complete red herring. But what we were trying to do in these early experiments wasn't to test orientation. Really what we were after is to demonstrate that you could measure connectivity in vivo, that you could put a coil in, stimulate an area, and see what it connected to. So basically treating the brain as a circuit board, and if we stimulate here, will we hit these other regions? Will we see current spread or activation of these other regions, not by volume conduction, but by pathway conduction? And the answer was yes, and I apologize for the quality of this. This is what you can get from NeuroReport, and my old PowerPoint files, my new PowerPoint won't read. And so this is what I can get off NeuroReport site until I get my files rescued. But basically what it showed was when you stimulate M1, you get S1, you get M2 and S2, you get SMA, so definitely you get a network effect. The other thing that was quite clear is even though we'd been told to expect that this is going to be big, okay, this isn't big, this is very, very focal, and it's not at the gyrus. It's deep. It's basically right at M1. So when you're stimulating, and we did record here, we recorded EMG, and so although this was handheld at this time, we were monitoring EMG and making sure we were getting FDI contractions throughout for each subject, and we did. So we knew we were hitting M1, and we're getting deep responses and very appropriate network-based responses. We're getting contralateral inhibitory effects, which EMG absolutely confirms. So we knew we were doing something right, and so we scratched our heads about this for a while and then went back and found all those early papers and digested them and tried to figure out what this actually meant. And we came up with a few things. First, we came up with the notion following day that this is column-based. But when we started modeling it and realizing what that meant, that it was column-based, we realized that this absolutely has to be done in simulations. So you have to actually model the magnetic field, you have to model the cortical surface, you have to make a per-subject model to have this information be useful. And the precision of that was such, we considered armatures and we measured their sag and found that these suck, that there is just no earthly way. And so we kicked out a bunch of money to get an FDA-approved neurosurgical robot and got an agreement from the company that we could reprogram their robot to do what we wanted to do. So we got a developer's agreement to reprogram their robot and built a completely robotic system that was also MRI compatible. This is for during the sham, that it makes a clicking that's at the same decibel level relative to the ear as this. And so this was the experimental setup that we used to acquire these data. And we were looking, so this basically shows you, in each person we did PET blood flow activation of finger tapping, and then we also did TMS getting FDI contractions, and so these are matched. So it's clearly deep. It didn't gyral. This is very much sulcal activation. And across subjects, we found that wherever we got the hand movement response in terms of depth from the surface, that's where we got the TMS response. So the idea that you could only stimulate near the coil and that would be the driving factor, it's like, yeah, so that's certainly not true. Driving factor is where M1 is and what the orientation is. It doesn't have much to do with how close or far you are from the coil, with one exception. That is, so this is modeling the coil, and right near the surface, the absolute E field is the most. But since on the gyroconvexity, the neurons are oriented this way, so the effective E field, which is the cosine of the angle between the long axis of the neuron and the E field. So here it's 0. So you get no stimulation, and that agrees with the fact that we didn't see anything at the gyri. And as you go down, you get more and more potential. When you really get right down the axis of the neuron, even though the absolute E field is about a fifth, so it's 100, but it's 100% effective. And so when we computed the effective E field, we got numbers that were very consistent across subjects. It's 80 to 100 volts per meter. And that's what we got to stimulate people. So this notion that it's the column or the cosine to the cortical column, that's how you compute what you're going to stimulate, gave us a way of figuring out for any brain area, could we stimulate it or not? So some areas we can't stimulate. We can't stimulate gyri, but we can figure out what our effective stimulation is at any given point. We turned our data over to the group, David Mogul's group, at University of Illinois, Chicago, and his postdoc was Todd Creed, and we just gave him the data and said, you guys are electric field modelers, knock yourselves out. And so they modeled it their own way and figured out, so what the map is here is of the effective stimulation. So they wanted to independently confirm whether the cortical column cosine or C3 model agreed with the data. And their answer is, yeah, it agrees with the data, and that in general, the stimulation is going to be maximum when the E field is oriented along the column. So there have been other modeling efforts, and I'll come back to other validations of that. But one of the easiest to grasp validations actually published by Shalini Narayana. She'll be talking tomorrow. So this is stimulating supplementary motor area. And so for supplementary motor area, if you use the more standard orientations of forward or toward the nose, you either get no activation if you point it forward, or you get very weak activation if you point it at 45 degrees. And so the maximum activation here is 90 degrees. It's, as Shalini called it, which I think is cute, B-ear. It's not B-nose, pointing it at the ear, because that's when you hit the neuronal orientation along the cortical surface of SMA. And so what she's showing is TMS is in red, and finger tapping activation is in green, and the overlap is in yellow. And so basically you're getting the same area, but you're doing it by orienting the coil this way. And when you go this way, you get zip. And these are other papers. These are some of the most interesting that have used this orientation for doing things like speech interruption. Works quite well. You can build a model having people phonate, aim at the phonating area using this model, and you'll interrupt speech. So this has been used in humans. Felipe Salinas will be also talking tomorrow. He's been using this in baboons and using the same aiming principle to study parametric modulation of TMS in baboons. So the description of this method and the robotics was published by Jack Lancaster in 2004. The first generation neurosurgical robot is here. That's still actually up and running in our lab. That's what we use in the PET scanner, even though it's ancient. Robots do tend to last a long time. You can see we had to jury rig it to add enough degrees of freedom. We had to add more degrees of freedom at the end. And then this is a more modern robot that we're using in clinical trials now. So there's two components. There's treatment planning. We do it all using structural and functional MRI, and I'll show a little bit more of our current strategies at the end. So we modeled scalp, we modeled a cortical surface, We use the cortical surface to register 2 per subject. We have to identify the site, and there's more to say about selecting the site. Then we determine the best stimulation orientation using the E-field electromagnetic field to cortex dot product, create a plan. That plan is saved, and it goes to the robot, and then the robot delivers the TMS. And for the treatment delivery, what we do is digitize the scalp and then register the robot to the scalp, and then the robot positions the coil and delivers the treatment. So a subsequent paper is doing intensity modulation of TMS. This is in motor cortex. It's correlated with the MEP. So the PET response and the MEP response work together reasonably well. There's good electrophysiological data, particularly by Charles Capiday, who's one of the authors on this, that he's done nice work using MEP to show what the response should be. And basically it plateaus after about 120 volts per meter. I'd have to check that number, but it's in that vicinity, that it goes up, 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 and then tails off. It's more or less a sigmoid function. Okay. So now when we're doing this, we were seeing networks, we were seeing multiple regions, so we wanted to start building models of these, and particularly, instead of just doing interregional covariances in a univariate manner, we wanted to model the whole network using multivariate approaches. And so what we began working with several years ago, and we're still working with, is structural equation modeling. And so with a lot of what we do in terms of structural equation modeling, we start with a meta-analysis. And so in this paper, Angie Laird did 2 meta-analyses, one of finger tapping and one of TMS to M1 hand, and taking the coordinates and figuring out where the optimal response locations are, and then using those to identify what the remote regions are, so what regions should we include in the model. So these regions are specified by this literature meta-analysis rather than by the responses in our subject group. So we predefined which regions are nodes in the network. And so then this is using intensity modulated data. So this is per subject data where we've done TMS to M1 hand using the robotic system and driving at 75, 100, and 125% of motor threshold. In our current trials, we don't use motor threshold. We use E-field magnitude. That if we're stimulating DLPFC, who cares what the M1 hand motor threshold is? It seems ultimately irrelevant. What you want to know is what you're delivering at the site you're stimulating. And so this is done using exploratory modeling. This, from the coil to M1, we know because we're controlling that. So that path is the first step. We're assuming that all the current flows or the effect, the pathway-based remote activation is starting at M1. And if you do exploratory modeling, just seeding M1 and asking which of the other regions in the network are the primary responders, these are the ones you get: SMA, cingulate, S2, cerebellum. And if you take each of those and you seed them and continue doing exploratory modeling, you get which are the secondary paths, and you can do that up to tertiary paths. Dr. Laird stopped at tertiary paths because she'd accounted for all possible variants in the model, so not much point in going further. In general, what we're finding is that structural equation models built with imaging data account for way more variance with even very small subject numbers than the usual uses of SEM, which are often based on cognitive performance. And so we have collaborators who specialize in structural equation modeling, and they love the imaging data. They said, this is the best data we ever touch. And the power analyses that have been done show that you can get really excellent model fits with subject groups even under 10. whereas typically structural equation modeling takes 150 to 200 subjects to get decent model fits. So one of the problems that we've had, it's a nice problem to have, is that we top out on the model fit statistics with imaging data. That's, that's like, okay. So it's not that difficult to get perfect model fits. So it makes it a little hard to choose between competing models when you have a variety of model fits which are basically perfect. So we've started doing things like pruning the number of paths and reducing the models to fewer paths so that we have less good fits so we can discriminate between different models. So given the idea that we can do parametric manipulation, so that was using intensity, another way of doing parametric manipulation, which is very useful for driving the models, is to do rate effects, frequency effects. And so Felipe Salinas, Shalini Narayana, and others have worked on this. This actually shows the current generation of our treatment planning software, and so it's modeling the coil. You can try different positions and orientations and calculate what the effective stimulation should be. This wasn't done robotically. For the monkeys, it's a little hard to get the robot and the scanner all to work together, but the locations were and the orientations were stored and using this system to stimulate. So there's a baboon in the scanner. Baboons are nice, actually. Their heads are big, their brains are big, bigger than rhesus. You don't get into the same IACUC problems if you try and do this on chimps. We've scanned chimps, but it's a big hassle to get approval to scan chimps. And so we think baboons are really quite a nice non-human primate model for developing TMS and other forms of electrophysiological stimulation. And Felipe, talking tomorrow, is a wealth of information about that. He's been doing pretty much, spearheading pretty much all of the non-human primate electrophysiology work in our group. So what's shown here in this rate dependency, if you take all frequencies versus rest and just make a map of what stimulation across a band of frequencies gives you, we couldn't really do meta-analysis in the baboon. There's nothing published in the baboon about M1 or TMS. So instead we did all TMS versus rest and identified again the regions of the model. So these show the frequency effects at different brain regions. And first thing to note is that not all regions are the same at where they peak. So it's not that there's one optimal frequency. And yeah, so there is a frequency dependence. And so the notion that, well, slow is inhibitory and fast is excitatory, it's like, okay, that's okay, but crude. There are definitely frequency-dependent effects that need to be mapped much more thoroughly, and it looks like by frequency you can pick pathways. You can optimize for specific pathways based on frequency dependence. Some pathways are excitatory, some pathways, like to the cingulate, are inhibitory, to the thalamus, inhibitory. So you need to know the sign as well as the frequency specificity of the pathway that you're attempting to stimulate. And using a very similar approach, Felipe is done, and this is not yet published, but I think all the analysis is done. The modeling looks pretty much done, so you can ask Felipe when he's going to submit the paper. But this is doing the same kind of strategy of starting the model with the rate effect onto M1 and then the primary, secondary, and tertiary paths. And here it's been strongly filtered for the strongest paths, and these are very strong path coefficients, again with no residual variance. That even with a relatively simple model, all the variance has been accounted for as best as the modeling software can see. I won't go through this in any detail other than to show that the rate-dependent effects and the path coefficients vary by the stimulus frequency. So whether you're getting primary, secondary, or tertiary pathways will vary depending on the stimulation frequency. Overall, so if you want to just take a big message, if you want to drive this motor system, drive it at 5 hertz. That's the easiest way to get most of the motor network. So now a bit of a detour. The reason for the detour is to show you where we're going in terms of identifying sites for stimulation. And so what we use for this approach is based on Brainmap. So Brainmap as publicly accessible data. It's all coordinate-based data as published. So literature data. It's got 2 sectors to it. There's a functional sector which has, at this point, about 12,000 experiments, and that's just kind of steadily going. We have 4 people who full-time code experiments to enter into the database. There's a structural sector, which is still only about 3,000 experiments, although that's also growing fairly rapidly. So the database has in it this much data, or did as of 6 months ago. You can search through it using Sleuth. It's entered using Scribe, and the statistical analyses, the meta-analyses are done using gyral. At this point, this is from January. It's now over 500 publications that have used this environment, the software and the data. And so they're doing a variety of different types of analyses. The most common are just either functional or structural meta-analyses. Given this task, which brain regions are most reliably activated, or given this disease, which brain regions most reliably show voxel-based morphometric assessed atrophy. But there are other things you can do with it. And so these take larger scale access. These are essentially modeling the entire database at once. So it's all viewed as one experiment, and each experiment within this one experiment is viewed as a moment in time. And so we're doing analyses on the functional sector, okay, in which we consider 12,000 instances, so each experiment is a moment, the way you would take each scan in a resting state study as a moment across a time series. So we're viewing the database in the same way. So when you do this, you see large-scale co-activation patterns that are quite robust. And they're very similar to what you get out of resting state. I'll show you in a moment how similar, but it's very similar. It's not identical, but it's really quite close. And so the first paper to do this was published by Roberto Toro in 2008, and he seeded each region and saw common thing in resting state is you get out the homologous region very nicely. So if that's what's shown here, these are the different region seeds that he used, and then these are the responses, co-activations, and Roberto said in the paper, basically, these look a lot like resting state, but he didn't validate it against resting state. So that left the window open for Steve Smith. Roberto was part of this as well. But what was then done, and is shown here, is on the left of each panel is an independent component from resting state, and on the right is the same software, same analysis, exactly on the co-activation data. So one is entirely resting data, so the left panel each time, and one is entirely activation data. And you can look at them and see how similar they are, but it's extremely similar. So that means you can use, instead of, this was validated with 35 subjects, you can use not 12,000 subjects, you can use 12,000 experiments to do basically resting state co-activation. So the volume, the clarity of the data from this is just overwhelming. So if you want to map pathways, this is the way to do it. You can get really very, very precise about what projects to what by using co-activations. So we do this really quite a lot. And there are some wrinkles on this that you can seed specific regions and get out what are co-activation maps for the region, but the database also contains the paradigms for all of these, as well as the behavioral domains engaged by the tasks. So you can actually get out pathway-specific or behavior-specific pathways. So you can say, okay, I want to know what the caudate connects to, but I want to filter it by what you're doing. And so you can see which of the caudate's regions are involved in cognition, which in perception, which in emotion, and which pathways follow which of these different behavioral domains. Okay? You can do this as well for paradigm classes, but that shrinks the data volume. So there are ways now of getting quite specific about pathway using very large data sets. There have been a lot of validations of this against resting states. So the most common thing that we're doing now is doing meta-analytic coactivation models, and with a conjunction with resting state, and often then with a confirmatory experiment using DTI. So this paper was done using DTI confirmation. This one by Danilo Bizdock is really interesting. They did the validation using 25 postmortem brains and did cytoarchitecture and did clustering analysis on the cytoarchitecture, and so that's shown on the left to show how cytoarchitecture segments within the amygdala, and then what's shown on the right is a cluster analysis where they did voxel by voxel connectivity of the amygdala and sorted the amygdala voxels based on their connectivity to see which ones co-activated in a coherent manner and compared that to cytoarchitecture. It's like, so I don't know if it gets much better than that of validating your connectivity maps against cytoarchitecture. Maybe a little bit better. So this is by Shalini Narayana, in which she's done a AL meta-analysis here, identifying regions of interest for the SMA, and then she did TMS PET to demonstrate which regions are activated or co-activated with SMA when you actually fire them with TMS. So this paper came out 3 years ago. And then this is showing, this radial diagram is showing the meta-analytic connectivity versus the TMS PET connectivity, and where they're similar and where they're different. So they're not identical. For the TMS PET, you're going to get weighting toward what's the fewest synapses away from the stimulated site, which is M1. And for the co-activation, likely you wouldn't get that. So how do we bring this back to what Vince started us with, which is how do we get it in the clinic? And so we've been thinking about that for a good while and trying to figure out the path to actually get it into the clinic and have a reasonable strategy for deploying all of this information per patient. So I think it's been known for a good while, and the early papers out of the NIH with Mark George, Eric Wasserman, and others basically arguing that depression is a network abnormality. There are functional abnormalities that are very nicely described by imaging, so you know it's multi-regional, that it's network-based. You can hit a limited number of these regions with TMS, so pick a region to stimulate, but don't expect that that's the only region you're hitting. Just realize you're tapping into a network. The reason they went to DLPFC, it's reliably implicated in depression, and you can hit it. So how did they choose to hit it? They actually went back to those same papers of Brazil Neto and Mills, and they said, well, We can hit M1 reliably by aiming at the nose, and we know that DLPFC, based on the Taylor-Ack atlas, is more or less 5 centimeters in front. So we'll just take the coil aimed at the nose and move it 5 centimeters to the front, and that ought to do it. It's like, wait, that assumes that you have another sulcus that's oriented just like the central sulcus, 5 centimeters in front, which you don't, right? So that's a bad idea. On the other hand, it worked reasonably well, and I think one of the reasons it works really reasonably well is that the gyri are all really confused. So you're going to hit something, and it will be within DLPFC. Okay. So, but maybe there's a better way of thinking about this. And this was clear. This is the O'Riordan trial that was the trial that FDA approval of this modality is based on. And he just says, we did it this way, but in the discussion he said, yeah, we're fully aware that this isn't the best way, but this is the way we've got. And image-guided and MRI-guided neuronavigation would certainly be better. Go at it. It's like, all right, that sounds like an invitation. Do something better. We admit that this isn't super. And one of the authors on that original O'Riordan paper, Paul Fitzgerald, then did image-guided and published that about a year later, showing that image-guided was better, but this didn't change orientation. It just it found DLPFC better. So it got him in the ballpark better, because it's not just 5 millimeters, it's 5 millimeters more or less. And so he did that per subject and got an improved response. But what we would appeal to is a strategy that was put out by the last author on this paper is Helen Mayberg, who's been a big motivator of brain stimulation and depression. So what they argued was that you need to know the connectivity of the region you're aiming at. Subgenual cingulate is the region that is most reliably implicated in depression. DLPFC projects to it, other areas mid-cingulate project to it. So let's figure out where we want to stimulate based on what will get to subgenual cingulate. And so this used DTI tractography to identify which regions were stimulation candidates and did a clinical trial. So what we would argue is a little bit differently. This is, we're not doing deep brain stimulation, so we don't have any target. We're still under the same constraints that Mark George was, which is that DLPFC is an easy area to hit with TMS. But how do we hit it well? And so this is a meta-analytic connectivity map doing a connectivity-based parcellation of DLPFC. And so if you take DLPFC more broadly, and then you do hierarchical clustering, you do per voxel connectivity, and you look at which voxels coherently project, the anterior portion of DLPFC, anterior and more medial, more reliably projects to subgenual cingulate, whereas the lateral and posterior portion projects to mid-cingulate. Now, mid-cingulate does project to subgenual cingulate as well, but the most direct projections to subgenual cingulate are from here. So this would suggest that finding this region per subject and aiming at it should increase efficacy. So that's what we're in the middle of doing now, is doing per subject targeting based on that meta-analytic approach. And so overall conclusions for this, that the local effect of TMS measured by PET certainly informs the neurophysiology, that is telling us that, yeah, forget the gyri and forget horizontal interneurons, that's just wrong. That it's hitting sulci, it looks very much based on the long axis, so it's following the column orientation. But beyond that, so once you get that down, that it's driving integrated networks, that the integrated networks agree with our other network modeling approaches pretty well. The aiming methods that are implemented clinically are neurophysiologically unsound and technically primitive, but cheap. Robots are not cheap, but we certainly need to move forward to clinical trials that aim better, that deliver the stimulation more reliably. So we really do think robots are going to be needed, and that the treatment planning should be informed by connectivity at an individual level. And this should lead to, I think, a new generation of TMS paradigms. And so that's where we are. And I'll stop there.